Okay, great. So thank you for the invitation to talk to you about some of the work that comes from my lab. As I was said, I'm professor at Johns Hopkins University and director of the RNA Innovation Center at Johns Hopkins. And I'm going to tell you today about work from my lab on mRNA metabolism and various methods that we're using to develop precision therapeutics specifically for haploinsufficiency disorders. So in my lab, we work on mRNA, and we worked on mRNA way before anybody really knew what mRNA was, more broadly in the community. And we think about mRNA very differently from, I think, what others do, in that mRNA is born in the nucleus, and then it has to traverse into the cytoplasm. And the function of mRNA is to uh, provide information from DNA to ribosomes so that they can then make a protein product. So the function of an mRNA basically is to encode a protein from the DNA. Now, what's important here, though, is that mRNA is really will continue to translate forever as long as it exists within the cytoplasm. And so that if you get a signal that stops the production of the mRNA in the nucleus, that signal is pretty much irrelevant as long as this mRNA is living in the cytoplasm and undergoing mRNA translation. And so the cell has decided or evolved basically that every mRNA is programmed to be destroyed. And these are destroyed in very defined ways where each mRNA has a predisposition to be destroyed at a certain rate. And so, for example, I'm just showing that this mRNA is destroyed within 30 minutes. And this allows the cell to control a linear flow of genetic information from DNA to protein uninterrupted and maintain that there is no dysregulation that would occur within the cytoplasm. And so my lab has studied that process of mRNA decay, and we're using what we know about mRNA decay to leverage how we would impact dosage disorders such as haploinsufficiencies. And the premise really here is that we know in a haploinsufficiency, one of the two copies of DNA has been mutated such that half the amount of protein is present. And so that can lead to a decrease in the amount of that mRNA or the production of a variant mRNA or protein. And so what my lab has decided to look for are technologies that we could use to work on the wild type copy of the mRNA in order to boost its overall translation rate and thereby restore the amount of protein product that comes from that gene. And so what we need to know for the rest of the story is that the way in which this programmed destruction of mRNA occurs is through this feature on an mRNA called a poly-A tail. So every mRNA receives this little tail at the end of it following its synthesis. And all you really need to remember here is that as long as this mRNA has a poly-A tail, it makes a protein. And that this poly-A tail is removed over time so that it's then lost. And when you don't have a poly-A tail, you don't make protein. And so poly-A plus, protein, no poly-A, no protein. And as I said, the way in which the cell controls how long an mRNA is stabilized or expressed in the cytoplasm is through progressive loss of this tail by these enzymes that I've drawn here as Pac-Man that are removing that tail at a defined rate. And then that leads to repression or degradation of that mRNA. So with that information in hand, how could we leverage that to create therapeutics that might be able to boost the expression of an mRNA? So what we've done is we have a very simple idea where we simply add an antisense oligonucleotide that's specific to the mRNA in question, but we polyadenylate it. So we add a poly tail to that, and we can modify this poly tail so that it cannot be removed by this Pac-Man. And that essentially has the feature of recruiting the proteins that normally bind to this tail, augmenting the expression in trans and allowing the mRNA to continue to be produced. And this is really work that goes way back to when I was a graduate student in the 90s, where I had shown that the protein that's associated with this tail can be recruited to the mRNA through pretty much any way you want, and it'll still stimulate the overall translation of this mRNA. And so we call this technology poly-A tail mimetic, or a mRNA booster. And we've been developing this to treat various haploinsufficiencies. And so this is, the general premise is you have an antisense guide sequence, which is similar. You can think of this as like an ASO, if you will. And then we put on a poly-A tail. We can modify this because these are relatively small. I'll show you sort of the size of where we can get down to toward the end of the talk. So we can produce chemical modifications in here that can help us increase the binding efficiency as well as improve the overall stability and efficacy of what we're looking at. And one of the attractive features of this technology is that every single mRNA in the human body, with the exception of a very small handful, which is really not therapeutically relevant, have a poly-A tail. So every mRNA in the human body is regulated by a poly-A. So in theory, this attachment of this poly-A tail mimetic to any mRNA could potentially enhance its overall expression. And so these are some of the first experiments we did with this technology, where we simply take an RNA oligo that we then polyadenylate using an enzyme called poly-A polymerase. And I can just show you this here, where we have the oligo that, sorry, Siri's talking to me. The oligo that we just buy, and then polyadenylate that, and you get a nice long tail on that. So this tail is about 300 nucleotides. And if we co-transfect into cells in vitro, just adding these poly-A tail mimetics with a reporter, this is just a luciferase reporter with some generic 3' untranslated region on that reporter, and then a control reporter alongside. You can see that when we add these poly-A tail mimetics, and these are just experiments done in quadruplicate or whatever this is, you get a nice robust increase in the overall expression of that mRNA in a specific manner. So in these cases, we were getting about a 3-fold increase in mRNA translation. Now, that was an in vitro experiment where we're looking at basically an IVT-derived mRNA. So this is an mRNA that we're introducing into the cell with the poly-A tail mimetic. What about endogenous, naturally occurring mRNAs? And so these are just 4 different experiments in vitro, again, in cell culture, where we're introducing a poly-A tail mimetic against mRNAs that are known to be associated with haploinsufficiency. And so the 3 are MECP2, which is involved in Rett syndrome, CTNNB1, which is beta-catenin deficiency, pure alpha deficiency, and SYNGAP1 deficiency. And in each case, we can generate a poly-A tail mimetic that will give us a significant increase in the overall expression of those mRNAs in vitro. And so this is a nice demonstration that the poly-A tail mimetic could be introduced into the cell and act on an endogenous mRNA, and in this case, a clinically relevant mRNA. So we spent most of our efforts working on SYNGAP1 deficiency, in part because SYNGAP was discovered by my colleague Rick Huganier at Johns Hopkins, and he's right upstairs from me, so it actually makes it a little easier to study for us. But I'm sure everyone's aware, SYNGAP1 is a developmental encephalopathy, and much like many of these other developmental encephalopathies like Dravet and Angelman syndrome, and basically leads to neuronal issues, including autism spectrum-like disorder, intellectual disability, etc. And like many of these encephalopathies, the big challenge that you have with developing a therapeutic is that they are the result of sporadic germline mutations. So they're not only very rare in occurrence within the population, but there's no real founder effect associated with them, that they are in fact random germline mutations. And so this is, I think there's about 1,500 SYNGAP patients worldwide that are known, and you can see there's 100 or so known mutations, pathological mutations, and so that makes it very challenging for developing therapeutics that might, for example, base editors or such that could be used against the gene of interest. So in this next set of experiments, we moved from just from cultured cells. These are looking in neurons, and you can see we can develop polyetail mimetics that will enhance SYNGAP1 expression at the mRNA level, and we get variable effects depending on the specifically polyetail mimetic that we design. In the case of SYNGAP, we've attempted to try to go through the untranslated. So I didn't say this, but every mRNA has a 3' region downstream of the actual open reading frame that encodes the protein. This is called the 3' UTR. These can be very large in some cases. So in the case of SYNGAP, this is about 1,800 nucleotides in size. And of course, the polyetail is over here. But what we've done is we've scanned through the 3' UTR to identify polyetail mimetics that have differential effects. And the reason for this is 3' UTRs are not barren. They are peppered with proteins and microRNAs and such that live in here and that control the regulation of the mRNA. So we want to make sure we get to regions that are accessible. This particular experiment was really, quite frankly, just a guess. We just moved through, and you can see we get kind of a nice histogram where this region here for this SB2 is the region that's giving us the maximum expression in these cultured cells for SYNGAP1 expression. And so we next wanted to turn our attention to away from in vitro experiments and look in mice and specifically in the brain. SYNGAP is expressed predominantly in the hippocampus of the brain. And so with a collaborator, we used a nanotube that we inserted into the hippocampus. And this nanotube has it's a stiff tube that allows you to wrap it around the hippocampus in this sort of like croissant shape. And then when you retract this nanotube, it leaves behind this super elastic and permeable membrane that we can then leave into the mouse and then pump and perfuse anything we want into the hippocampus. And in this case, we're putting in polyetail mimetics to look at SYNGAP expression. And this is our first experiment just looking at, this is just 3 mice. So there's no statistical power to this, but if you look at the, sorry, this isn't showing up very well, the black here, but you can see that in the control, when you look at SYNGAP expression, you get this basal level. But when you perfuse in these 2 polyetail mimetics, you get a significant increase in the hippocampus that's reproduced in the cerebral cortex and in the midbrain area. But you see nothing in the cerebellum where there's very little SYNGAP expression anyway. And this is just quantitation of those results. This is a repeat of that experiment using more mice that are both males and females. And you can see in our control, their normal expression of SYNGAP. And I should just mention these are wild-type mice. So this is sort of the wild-type expression of SYNGAP. And you can see in each of these mice, for the most part, we see a significant increase in expression of SYNGAP1 in the hippocampus. Now, because these are oligos, essentially like a ASO, what I've been showing you have been just naked RNA. So RNA is a very delicate molecule. It's degraded very quickly within the body. And so in order to use it as a therapeutic in any way, shape, or form, you have to go to chemistry that would both hide it from the immune system as well as protect it from degradation from various nucleases that exist within the body. And so in these experiments that I'll show you, we've used classic modifications that are known to improve the efficacy of ASOs. These are very similar to those that are used in, say, for example, Spinraza, where we have a 2' methyl group that will help block the oligos from being destroyed, and phosphorothiorates that essentially do the exact same thing that prevent endonucleotic cleavage of these oligos. And we also are putting them on both ends of the, only on the ends here of the oligos to look at the overall efficacy. And what I'm showing you here, these are actual northern blots of the polyetail mimetic where we've transfected. These are the Normal, the ones that I've been showing you already, and this is after a week of transfection, you can see what you really need to see is they're not there. They've been destroyed. But these version 3 molecules where we've introduced these modifications, we can significantly stabilize this after a week in culture. And these also give us a significant increase in expression of SYNGAP in cell culture. We've moved on from here to try to move toward a more a molecule that could be a potential therapeutic. And one way to do that is to shrink the size of the oligo. And so in these experiments, what we have been doing is reducing the size of the poly A tail. And I'll just mention that the poly A tail, the only function of the poly A tail is to recruit a protein that is called poly A binding protein. And poly A binding protein binds every 25 A's. And so a poly A tail of 100 nucleotides has 4 copies of poly A binding protein, and then 3, and then 2, and then 1. And for the most part, we know you really only need 1 copy of poly A binding protein in order to stimulate the translation of an mRNA. And so this is just an experiment showing you that when we modify these oligos, the poly A tails in this case, it'll still bind poly A binding protein. Now, if you're not a biochemist, then no big deal if you're not, but the way dissociation constants work is the lower this number, the stronger the binding. And so, in fact, our unmodified bind, poly A binding protein reasonably well, but the fully modified and the 3' and modified actually bind poly A binding protein a little bit better. We don't know why, but they do. So that's really cool. And if we shrink the tail, we see that even these molecules that have only 25 A's are still stimulating SYNGAP expression to a reasonable amount in wild type cells. And so this is starting to get into the ballpark of a therapeutic that could be of a decent size. So in this case, this is only a 50 nucleotide sequence between that guide and that A25. And so we're getting into an area that potentially could be used much like any other ASO. These are some unpublished and preliminary experiments. All the experiments that I've shown you so far are looking at wild type mice. We were not ready to work on haploinsufficiency models. And I would say it's still premature days for that because all the effects that I've shown you are relatively mild, meaning twofold effects. We'd like to have that be a bit more robust within a mouse, just because you know that translating that to a human being is always going to take a hit. But we have started some work with Rick's lab on SYNGAP1 mouse models where in this experiment, what we did is we blindly injected P1 mice with poly A tail mimetics, and this was injected into mice that where we didn't know the genotype. And then we took down the mice after 20 days and then looked at SYNGAP expression and then de-identified which mice were which. And first thing I'll show you is that if you look at the weight gain of those mice over time, they're all pretty much normal. So we're not detecting any sort of toxicity, at least in this very crude assay with these mice. And we're getting a stimulation of SYNGAP expression. And this is a very, very small pilot experiment, both in the wild type as well as in the SYNGAP heterozygote that after we de-identified it. So again, there's a lot of work that has to go here to really optimize this. I can tell you, from my experience at Tevard, where we've started working on RNAs that we're developing for haploinsufficiencies. When we first started the company, this was the sort of delta that we were seeing for various mRNAs that we tested. And with over 7 years of work, we now have molecules that are getting 60 to 70-fold increases over baseline. And so it takes a lot of future optimization in order to get something, a signal like this, and produce it into something that's going to be clinically meaningful. But it's a start. And so what I've shown you today are, again, some preliminary uh, uh, research that we're doing in the lab to try and leverage our knowledge of mRNA metabolism to develop drugs that could be used to treat dosage disorders, haploinsufficiency disorders. And we've shown data I'm not showing you today. We've shown this in three different mouse models, where if we tether a polyetail mimetic to the 3' end of an mRNA, we can get an increase in the expression of MECP2, of beta-catenin, and I've shown you, most of what I've shown you is through SYNGAP1. But in these other cases, we have a significant amount of data, especially with beta-catenin, that we can in fact enhance beta-catenin levels and demonstrate target engagement in that system. So in summary then, we've developed a novel gene therapy that enhances protein expression. It's a first-in-class technology. It's very preliminary in its development. It leverages the power of RNA therapeutics. We would hope that it would be disease-modifying because it works at the mRNA level and would enhance the expression of the wild-type mRNA. It is presumably mutation agnostic because it can work on the wild-type mRNA. I should say that we have no ability to discriminate between the diseased mRNA and the wild-type mRNA, but as long as the mutant copy is producing a protein that's non-functional and is not dominant negative in any fashion, it really wouldn't matter. So increasing the expression of a barren protein twofold or threefold really would have no consequence. And lastly, we think this is a platform technology because, as I mentioned at some point in the talk, that most human genes, 98% of human mRNAs have a polyetail, and that polyetail is critical for their expression. So porting this across different indications is something that seems feasible. And we've shown now for about half a dozen mRNAs that we can enhance their expression using this technology. And so with that, I'll just conclude. And these are the people in my lab, past and present, and many of our collaborators that have helped along the way in these projects, and people that have given us funding. I didn't have time, sorry, but we're in a new initiative here with Combined Brain to work on several of these haploinsufficiency genes to see whether this technology could be used in those cases. And of course, these are the members of the RNA Innovation Center that work on mRNA technology. So with that, I'll stop, and I believe we have some time for questions. Thank you.